Thank you everyone for attending. My name is Jan Schellenberger. I'm a tech lead for the classification infrastructure team at GRAIL Bio. I want to tell you about how we do machine learning at GRAIL. First, there's some disclaimers from GRAIL. At GRAIL, our mission is to detect cancer early when it can be cured. As you may know, cancer is the #2 cause of death in the US following heart disease. The economic cost is estimated in the hundreds of billions of dollars per year, and the human cost incalculable. A lot of effort has been spent on improving cancer treatments, but we could actually save a lot more lives if we could just detect the cancer earlier. That's because if a cancer is found before it has spread to other parts of the body, it can often be removed by surgery, and the 5-year survival is close to 90%. On the other hand, once cancer has spread, surgery is usually not an option, and the only treatments are various poisons, which happen to be slightly more poisonous to the cancer cells than to the rest of the body. In other words, chemotherapy. The 5-year survival for metastatic cancers is only about 20%. These statistics are slightly misleading since the cancer makeup of the left and right panel are not identical, but the trend is correct. For any cancer type, 5-year survival gets worse once the cancer spreads. There are a few cancer screens currently available. For example, mammography for breast cancer, pap smear for cervical cancer, colonoscopy for colon cancer, and low-dose CT for lung cancer, but only if you've been a heavy smoker. For most types of cancers, there are no screenings available, and these cancers are found when they present symptoms or simply by luck. The sensitivity of all these screens is quite good, catching close to 90% of their respective cancers. However, the low specificity and low prevalence of cancer means that most positive test results are actually false positives. This is reflected in the low positive predicted value of these tests. If you want to detect several cancers by doing several individual screens, the false positive rates stack. A 60-year-old female with a history of smoking may get a low-dose CT to screen for lung cancer, a pap smear for cervical cancer, a stool-based screen for colon cancer, and a mammogram for breast cancer. Between these 4 tests, she would expect a 37% chance of having at least one false positive requiring follow-up. GRAIL is trying to build a cancer screen to supplement the existing screening modalities that can detect multiple cancer types from a single blood draw. What are the requirements for such a cancer screen? Number 1, it should have a low false positive rate. Every positive test needs to be worked up, causing medical risk, cost, and anxiety. In a target population of retirees, it is estimated that around 1% of patients would have a cancer. So a screen with 95% specificity or 5% false positive rate would mean that for every cancer we detect, at least 5 patients would receive a false positive result. This is actually considered a pretty good ratio in the screening world, but at GRAIL, we are targeting at least 99% specificity or 1% false positives. Number 2, it is not enough that a multi-cancer test tells you that you may have cancer. It should also tell a physician where to look. Is it likely breast cancer or pancreatic cancer or lung cancer? The workups for these different cancers is quite different, and looking everywhere in the body for a possible cancer is risky and expensive. Finally, a cancer screen should catch the clinically dangerous cancers. Overdiagnosis, where patients receive unnecessary treatments for benign conditions, are a real problem. So this multi-cancer screen is GRAIL's goal. To get there, we are developing a DNA-based blood test to detect more than 50 deadly cancers with high specificity. We are running several large-scale studies to provide evidence that this test works and saves lives. And we use technology, including machine learning, to drive test performance and gain new biological insights. Cancer is a disease where DNA mutations cause a cell to replicate uncontrollably. Sometimes when these cancer cells die, they release their mutated DNA into the bloodstream where we can detect it with a blood draw. The fraction of circulating DNA coming from cancer cells is tiny, so detecting cancer is a needle in a haystack problem. Some of the kinds of mutation signals that have been studied at GRAIL are, number one, small point mutations in known cancer hotspots. You may have heard of the p53 gene. Many cancers develop mutations there. If you happen to find a DNA fragment with such a mutation, it may have come from a tumor. Number two, large chromosome alterations where whole chunks of genome are duplicated or deleted. These show up as spikes and dips in a coverage plot. We find too many or too few DNA fragments from a particular region of the genome compared to what we expect. And finally, there is a process by which DNA is chemically modified or methylated. DNA methylation is one of the ways that a skin cell is different from a liver cell or a neuron, despite the fact that they all have the same basic DNA. DNA is composed of a sequence of 4 bases, usually abbreviated A, C, T, and G. In mammals, when there is a C followed by a G, the C can have a methyl group attached. In order to sequence or read these methylation patterns, we use a chemical process which converts all C's to T's, except if the C has a methyl group on it. So when we read the DNA fragments and find a T where there should have been a C, we know it was not methylated. If we find a C, then the DNA was methylated. GRAIL is running numerous clinical studies. In the Circulating Cel-free Genomic Atlas, or CCGA study, we enrolled about 15,000 participants, some with diagnosed cancers and some healthy with no known cancers. We collect blood samples from these participants and track them to see how they progress. Phase 1 of this study was exploratory, and we tested the 3 different cancer signal approaches. For phase 2, we have selected the methylation signal and are now training a full classifier on larger data set. For the rest of the talk, all the data is from the CCGA Part 2 study. At a high level, GRAIL's approach is a classic machine learning problem. We have training examples consisting of blood samples from healthy participants and blood samples from patients with various kinds of cancers. We use laboratory techniques to process these samples and then sequence or read the DNA fragments that are floating in the blood plasma. After running the raw data through a bioinformatics pipeline, we extract numerical features and apply standard machine learning algorithms to produce the GRAIL classifier. One aspect that is quite different from most machine learning approaches, the process of going from blood tube to numerical features involves both a lab component and a software component. We can always change the software, but the lab procedure has to be fixed beforehand, and once the procedure is locked, we usually cannot change it. Planning is key. There are several challenges to doing machine learning for early cancer detection, and I'm going to highlight some of them. The first challenge is, how do we go about collecting samples? The cancer samples are relatively straightforward. When a patient is diagnosed with a cancer, we ask for permission to receive a blood sample along with some other information. There are some efforts around finding particular rare types and stages. Getting a clean set of non-cancer blood samples presents a challenge. As I've mentioned, we are targeting a false positive rate of less than 1%. This is close to the rate of undetected cancer we would expect in an older population, but there is initially no way to know who the 1% with cancer are. I mean, if we could tell who has cancer, we wouldn't be in this business in the first place. This means that a lot of samples that look like false positives to us are likely cancers that just haven't been discovered yet. Now, we could try to collect blood from younger, healthier population where cancer is much rarer. But then we would be building a classifier that can differentiate between blood from young, healthy people and blood from older cancer patients. It turns out it's much easier to tell the age of a patient from their blood than to detect cancer. The only solution is to collect blood from an age-matched population and then wait 12 or even 24 months to see if any cancer develops and then exclude those samples. This means that 1, we need to have follow-ups with our study participants, and 2, in a machine learning sense, our training population is changing over time. We may have several interactions with a single enrolled study participant. The initial enrollment would be recorded into our electronic data capture or EDC system, including cancer status, age, sex, some demographics, smoking history, etc. Special attention is paid to protecting personal identifiable information, which we never use for any analysis. A blood sample will be sent to our lab, cataloged, sequenced, and processed through a bioinformatics pipeline. At a later time, clinical follow-ups may reveal a change in cancer status. This would also be captured into our EDC. We may receive a tissue sample from participants with a solid tumor biopsy. This means that our understanding of our trial participants is changing over time. We have built a special service that provides us with snapshotted versions of our training data, which we refer to as tidy data. When we have a major change on the clinical side, or we release a new bioinformatics pipeline, we create a new version of our tidy dataset. This provides a convenient way for different modelers to refer to exactly the same dataset. Anyone who has worked with real-world machine learning data knows that data cleaning is extremely important. Because we rely on different outside medical providers, there is opportunity for errors to creep in. These include simple typos, missing data, etc., but also clinical mistakes where we see a reported cancer type that is not consistent with other data. We have a team of clinical data scientists who spend a lot of resources following up and fixing these kinds of issues. Many applications of ML have a problem of label noise, where labels assigned to your data is ambiguous or wrong. At GRAIL, label noise comes in many forms. We may find out that a participant now has a diagnosed cancer and likely had that cancer at time of the blood draw. The boundary of what is considered cancer is also not clear and shifting. There are numerous precancerous conditions that share some characteristics with cancer, but may increase the chance of cancer in the future, but technically are not cancer and may not be associated with increased mortality themselves. Sometimes these conditions get reclassified by the medical community. Cancer types are also hierarchical. For example, there are at least 3 major subtypes of lung cancer based on what kind of cell the cancer originated from. Breast cancers are categorized by type of receptors they present and what hormones they respond to. This hierarchy is an important factor to consider because the classifier may be able to tell some subtypes apart more easily, or it may preferentially confuse one subtype with another cancer type altogether because they share some biology. We need flexibility in how we assign our final training labels so they work best with our classifier and are most useful in making a diagnosis. Some other unusual events that can happen. There are advanced metastatic cancers where it is unclear which organ they originated from, and some patients have multiple cancers at the same time. And finally, there is a long tail of rare cancers for which we may only have 1 or 2 examples. Here's what I believe to be a fairly typical machine learning flow. A training set of representative examples is collected. After some experimentation, a classifier is trained. Then, to provide proof that the classifier works, an independent validation set is collected. We test the classifier on the validation set and report the results. One of the biggest no-nos is to use insights from the validation set to update the classifier. This would bias the new classifier's results on the validation set and essentially invalidate the validation set. But there's another way to abuse the validation set, and that's through p-hacking. It's been said that if you torture the data long enough, it will confess to whatever you want. Similarly, if you can make your final data evaluation look better if you exclude that small population where it just doesn't quite seem to work. Think patients with a previous history of cancer or patients with a particular blood disorder. Or maybe you can combine those two labels that seem to be getting mixed up more than you anticipated. In a clinical trial setting, none of this is allowed. You have to pre-specify exactly the population you will look at and the metrics you will evaluate on. Here's roughly the same flow at GRAIL in a clinical setting. For the CCGA2 study, we collected the training and validation samples at the same time. To prevent anyone accidentally looking at the validation samples, a blinding protocol is set up with user-based access controls. We continue collecting clinical follow-up information, and at some point there is a clinical data lock which officially snapshots the data. No more changes to the clinical data set are possible. At this point, the official classifier can be trained. Before the validation set can be inspected, the classifier must be locked. The statistical analysis plan, or SAP, for evaluating the validation set must be approved and locked as well. This includes specifying exactly which populations will be included and excluded, which cancer labels will be evaluated against. The final report is written to reflect the format of the SAP. Only then can the blinding process be lifted and the validation set be evaluated. The final report is purely mechanical process, as all relevant analysis must be pre-specified. A few more words about blinding. As much as possible, GRAIL employees only see the parts of the data that are relevant to their role. The clinical data team, which is responsible for collecting the most accurate clinical information for each participant, cannot see any results from any classifier. This is to avoid bias. It would be tempting to resolve some ambiguity in cancer label based on the classifier's predictions. Likewise, the employees working in the wet lab to develop our procedures are not allowed to see any clinical information about the samples they are processing. Model developers are blinded from the wet lab metrics and have no say into which samples are considered evaluable. There's been much discussion about the so-called reproducibility problem in machine learning. Because we work in a regulated environment, this is particularly important to us at GRAIL, and we have safeguards in place to ensure we always know exactly how we ran every classifier. Each training and prediction run starts with a configuration file. We use protocol buffers to define these, but you can think of them as a JSON file. These configuration files define a training data set from our version data set and any population filters. They define how cancer labels are to be generated. They define the cross-validation strategy and any stratification. They define which training method is to be used and any parameters the training method may need. Finally, a random seed is set which controls the splitting and model initialization. After training is done, we save models and results in a standardized format. Additionally, we store the user ID, timestamp, configuration JSON, and Git version. If there are any local changes in the source code, we make a note of that too. In general, local changes are allowed only for experimentation. For any official run that is presented in a group meeting and above, local changes are not allowed. All code needs to be peer-reviewed and committed. We have built an evaluation framework that can coordinate all these parts. The evaluation framework takes as input the configuration JSON file. It talks to the tidy data server to obtain the correct version of the data. The framework then applies the correct population filter and label filters and splits the data across validation shuffles. It repeatedly calls the machine learning training and prediction process and stores the classifier scores along with the metadata. The scores are in a standard format, and we have tools for generating reports and key performance metrics of the classifier. By default, we do a determinism test by training and scoring one data shuffle twice and comparing the results to ensure that they are identical. A little bit about our classifier. Our current classifier works in two phases. In the first phase, we build region-specific DNA fragment models that can assign a probability of non-cancer and cancer type to each DNA fragment based on the methylation pattern. We keep track of outlier fragments with particularly high cancer scores, and these counts from different genomic regions are the raw numerical values that go into our final multi-class sample level classifier. Here's another view of the classification process. Different genomic regions can detect different methylation patterns found in one type of cancer but not another. We binarize counts, and this is the input to a generalized logistic regression. There's a thresholding step that decides whether a cancer score is high enough to merit a positive call, and a calibration step that decides a tissue of origin. We have some interesting scaling challenges at GRAIL. The studies we have run so far have a training set of around 10,000 samples. This is not big by internet scale, but it's actually quite large for a biological study, and we anticipate much larger studies in the future. For each sample, we collect a huge amount of raw sequencing data, on the order of 10 gigabytes per sample. There's no way to do training on a single machine without serious compromises. So in general, we don't train on a single machine. Our classifiers get trained on in AWS on many large EC2 instances. We developed a system called Big Slice, which allows us to shard our data across different machines. Big Slice is similar to MapReduce, Hadoop, or Spark-like systems and has recently been open sourced. Technology is key to GRAIL success. GRAIL is active in the open source world. We have developed and maintained two large open source tools, Reflow and Big Slice, for dealing with distributed computing at different scales. We believe that without strong technology and infrastructure, we will be limiting ourselves in what kind of science we can do and how fast we can do it. One of GRAIL's founders and many of the initial software engineers came from Google, so a lot of our tech stack looks similar to Google's. Most of our systems are written in Go. It's time for some results. How does the GRAIL classifier actually work? This is a summary of the training and validation set of the CCGA2 sub-study. As you can see, the training set consists of about 3,000 participants, split evenly between cancer and non-cancer. The validation set is about 1,250 participants. Overall, we are able to detect 55% of the cancer samples in the validation set. This includes over 50 cancer types. When we restrict ourselves to a set of 12 types that we had previously selected before the study started, the detection goes up to 76%. These pre-specified cancers tend to be more aggressive cancers. You can see there is a strong stage dependence where later stages are much easier to detect than earlier stages, and stage 1 cancers in particular are quite difficult. At stage 2, on the other hand, we catch about 70% of the pre-specified cancers, and this is a very valuable time to catch cancer. The specificity achieved was 99.3% on the validation set, in other words, a false positive rate of just 0.7%. And this false positive rate is shared across the 50+ cancer types we can detect. This is an important point. If you were to hypothetically do 50 separate screenings, the false positive rates of these 50 screens would add together. We have a combined false positive rate of just 0.7%. Here's the same data broken down by some cancer types and stage. The number of examples is in parentheses. Many of these cancers have no screening available, so really any detected cancers are incrementally quite helpful. I'd like to highlight pancreatic cancer, which is the number 3 most deadly cancer, in part because there's no screening available and it is usually detected too late. The GRAIL classifier shows quite good detection even at stage 1. So that is the detection performance, but can we tell where the cancer signal is coming from? On the validation set, our classifier made a prediction of tissue of origin about 96% of the time. The other 4% of the time, the classifier is unsure and returns an indeterminate tissue of origin positive result. When we are able to make a prediction for tissue of origin, the top prediction is correct 93% of the time. And finally, I want to talk about what do we think would be the impact of deploying a test with these performance characteristics on an intended use population. We took data such as cancer incidence rates, cancer stage distribution, and estimated progression speed, and simulated what would happen if everyone between the ages of 50 and 79 took an annual GRAIL test. If we look at the population of people who have a diagnosis of cancer and die of this cancer within 5 years, this represents 350 individuals per 100,000 population. We estimate the GRAIL test could detect about 250 or almost 70% of these cases before they would currently be diagnosed. And then of these 250 cases that GRAIL test finds, we estimate that we would be able to find many of them at an earlier stage where their cancers may be more treatable. In fact, of these 250 cases, we estimate that 37% of them would now survive beyond the original 5 years of their original diagnosis. This represents about 110 averted annual deaths per 100,000 population. I want to emphasize that these results are from a simulation. The simulation has been reviewed by subject matter experts and is being prepared for formal publication. To put these numbers into perspective, the number of deaths averted by a GRAIL multi-cancer screen in our target age group in the US is greater than the deaths from strokes, diabetes, accidents, or opioid overdoses. So this is the mission of GRAIL, to make a real impact on the number of premature deaths from cancer. The discovery that there is tumor signal in cell-free DNA happened less than 10 years ago. In another 10 years, such screens may be routine. I'd like to thank everyone for coming to my talk and finding out more about GRAIL's mission and technology. We are always looking for biologists, bioinformaticians, data scientists, software engineers, and machine learning experts to join our team. And at this point, I'd like to open it up for questions. Thank you.